Hello, and welcome to the webinar today entitled Next Generation Targets for Immuno-Oncology. My name is Scott Schachtel, a product manager at Bio-Techne, and I'll be your host for today's event. I'm happy to welcome today's presenter, Anthony Persson from R&D Systems, a Bio-Techne brand. This webinar is brought to you by Bio-Techne. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, and Advanced Cell Diagnostics to provide the immuno-oncology research community with high-quality recombinant proteins, bioactive antibodies, cell culture media, immune cell differentiation kits, small molecules, and analytic instrumentation. Today's presentation will be given by Anthony Persson, a manager in bioassay development at R&D Systems, a Bio-Techne brand. Anthony received his PhD in cell biology and anatomy at the University of Arizona and has over 10 years of experience developing novel cell-based assays, including those focusing on novel immune checkpoint inhibitors. We invite you to ask any questions you have for our presenter throughout the webinar using the Ask a Question box just below the presentation screen. These questions will be asked to the presenter during the live Q&A session directly following the presentation. Outstanding questions will be followed up on offline. In addition, in the upper right-hand corner of the screen, you will see a tab called Resources. In that tab, you'll find an extra content that relates to the next generation targets discussed during the upcoming webinar. Thank you again for attending today's webinar. Tony, I'll hand over the presentation to you now.
Hello everyone, and welcome to our webinar titled Next Generation Targets for Immuno-Oncology. The use of immune checkpoint inhibitor antibodies has truly revolutionized cancer therapy over the past several years, and we are excited to present some of our novel research involving next generation targets for immunotherapy research with you today. First, I would like to start with a brief overview of immunotherapy approaches being used in the clinic today. Cancer immunotherapy is basically modulating the immune system in a way that helps a person's immune system better recognize and eliminate cancerous cells. Cytokine therapy, the oldest method of immunotherapy, is simply the administration of cytokines systemically. Cytokines like IL-2, IL-7, and interferon alpha are generally given to patients in combination with chemotherapy and radiation to treat cancer. Administration of these cytokines causes a systemic inflammatory response that has been shown to enhance the immune system's ability to fight cancer. Cytokine therapy's effectiveness is somewhat modest, and these therapies may have unwanted side effects. Adoptive T-cell transfer is an exciting new area of immunotherapy research. Basically, T-cells are removed from a patient, genetically modified, cultured in vitro, and ultimately returned to the patient. Chimeric antigen receptors, also called CAR T-cells, are genetically engineered T-cells that have been shown in clinical trials to be highly effective in the treatment of leukemias and other cancers. Adoptive T-cell therapy is a promising area of research, but will not be the focus of our webinar today. The last type of immune therapy we would like to discuss is antibody therapies that target immune checkpoint modulators. In 2011, the first immune checkpoint antibody therapy targeting CTLA-4 was approved by the FDA, followed by anti-PD-1 therapy approval in 2014. These antibodies both work in a similar fashion to block the inhibition of T-cell function elicited by tumors, resulting in enhanced T-cell killing of tumor cells. This webinar will focus on describing new targets for checkpoint inhibitor research. Here we show how these checkpoint inhibitor antibodies function during cancer treatments. PD-1 expressed on tumor cells binds to PD-L1 on T cells, resulting in the inhibition of T cell killing on tumor cells. Antibodies that block the PD-1-PD-L1 interaction between the tumor cells and the T cells results in de-repressed T cells that are better able to recognize and kill tumor cells. The mechanism of action of CTLA-4 blocking antibodies is very similar to what we described for the anti-PD-1, PD-L1 blocking antibodies. CTLA-4 expressed on tumor cells binds to B7-1 or B7-2 receptors on T cells, allowing the tumor to reduce the T cell's ability to kill the tumor cell. Anti-CTLA-4 or anti-B7-1, B7-2 antibody therapies inhibit the ability of tumor cells to inhibit T cell activity, resulting in enhanced T cell mediated killing of tumor cells. Next, we will present our research involving potential new targets for immunotherapy research. On this slide, you can see many of the immune checkpoint receptors involved in tumor cell T cell signaling. Many of these interactions are well known, with clinical trials already underway using antibodies targeting these interactions. At R&D Systems, we have the largest catalog of recombinant proteins in the world, allowing us to screen for novel interactions that might be interesting in the context of immune regulation. Our protein-protein interaction screens, in conjunction with cell-based experimentation, uncovered new roles involving the Butyrophilins, VSIGs, and LILRs in the context of immune regulation. We will go over this data with you for the remainder of this webinar. First, we would like to discuss the Butyrophilin family of immune receptors. Butyrophilins belong to the immunoglobulin-Ig superfamily of transmembrane proteins. The Butyrophilin family shows structural similarity to the extracellular domain of the B7 family of receptors that have been shown to be receptors for CTLA-4. Butyrophilins have been shown to regulate T cell activation and differentiation. Butyrophilins represent a new area of investigation for the design of future strategies in the modulation of the immune system. Next, we will discuss data involving our Butyrophilin 3A1 recombinant protein. But first, some background on Butyrophilin 3A1. Butyrophilin 3A1 is expressed by most human immune cell subsets including T cells, B cells, monocytes, dendritic cells, and natural killer cells, as well as hematopoietic precursors and some tumor cell lines. Butyrophilin 3A1 is a ligand that mediates T cell inactivation. The B30.2 domain of Butyrophilin 3A1 is a receptor for the intracellular phosphoantigen, leading to the activation of gamma delta T cells. The receptor for Butyrophilin 3A1 on T cells remains unknown. In this experiment, T cells were isolated, and the addition of a recombinant ECD Butyrophilin 3A1 Fc fusion protein was shown to inhibit anti-CD3-induced IL-2 production at 24 hours post-treatment. The proliferation of T cells is not altered when treated with Butyrophilin 3A1 after 24 hours. The levels of IL-2 in this experiment were measured using the human IL-2 quantikine ELISA kit. Interestingly, Butyrophilin 3A1 also inhibits human T cell proliferation at 72 hours post-treatment, as is shown on this slide. The cell number in each well was assayed using our Razazarin fluorometric assay. Next, we tested the effects of treating CD4 positive T cells with our Butyrophilin 3A1 recombinant protein. Here we can see that human Butyrophilin 3A1 modified T cell receptor signaling to inhibit FOXP3 expression and regulatory T cell development in naive CD4 positive T cells. This data suggests that Butyrophilin 3A1 inhibits naive CD4 positive T cell differentiation to Treg cells. This data was obtained using our FlowX human regulatory T cell multi-flow kit. This slide provides a summary of our Butyrophilin 3A1 T cell studies. Our data demonstrates that Butyrophilin 3A1 acts as a T cell ligand and it inhibits T cell activation. Treatment of human T cells with recombinant human Butyrophilin 3A1 inhibits anti-CD3-induced cytokine production and T cell proliferation in a dose-dependent manner. Recombinant human Butyrophilin 3A1 inhibits naive CD4 positive T cell differentiation to Treg cells. Currently, the receptor for Butyrophilin 3A1 on T cells is not known, but efforts are currently underway to uncover this new potential checkpoint target. We currently provide bioactive recombinant butyrophilin 1A1, 2A1, 3A1, butyrophilin-like 2, and butyrophilin-like 8 proteins. Recombinant human butyrophilin 3A2 will be available in the near future. As you can see in this product table, almost all of the butyrophilin proteins inhibit T-cell activation and will be useful reagents for investigating the mechanisms of T-cell activation in the context of immunotherapy research in the future. Next, we would like to transition to discussing our data involving Vista and VSIG3. This novel interaction was initially discovered in an ELISA binding screen at R&D Systems. First, some background on Vista. The V domain containing Ig suppressor of T-cell activation, Vista, is also called differentiation of embryonic stem cells 1, DICE1, GI24, and PD1 homolog. Vista is a 55 to 65 kilodalton type 1 Ig membrane protein. Vista is highly expressed on antigen-presenting cells and to a lesser extent on CD4 positive T cells, CD8 positive T cells, Tregs, and infiltrating lymphocytes. Vista is a co-inhibitory ligand for T cells. Vista Ig fusion proteins inhibit T cell activation, proliferation, and cytokine production. Vista functions as an inhibitory receptor on CD4 positive T cells. Vista null CD4 positive T cells showed stronger antigen-specific proliferation and cytokine production than wild-type CD4 positive T cells. Vista is a negative checkpoint regulator of T cell activation. The binding partners of Vista are currently unknown. Next, I will discuss some background information involving VSIG3. The V-Set and Immunoglobulin domain containing 3, VSIG3, is also known as BT-IGSF and IGSF11, was originally identified as a member of the immunoglobulin superfamily and was shown to mediate homophilic adhesion in a calcium-independent manner. VSIG3 gene is expressed in the testes and ovary and at lower levels in the brain, kidney, and skeletal muscles. VSIG3 expression is elevated in colorectal cancers and hepatocellular carcinomas as well as in intestinal-type gastric cancers. VSIG3 regulates synaptic transmission and plasticity through interaction with the postsynaptic scaffolding protein PSD95 and AMPA glutamate receptors. To identify Vista interacting partners, we screen Vista binding with B7, Butyrophilin, SLAM, and VSIG family members using an ELISA binding screen. Under our experimental conditions, we did not find any evidence for the Vista protein binding to known B7 family members. However, we identified VSIG3 as a binding candidate of Vista. After our initial ELISA binding screen to identify Vista binding partners, we conducted experiments to confirm if Vista is truly a binding partner for VSIG3. Here we show ELISA binding data on the left, which demonstrates Vista binding to VSIG3. To further confirm the specific interaction between Vista and VSIG3, we carried out ELISA binding assays in the presence of sheep or mouse anti-human Vista blocking antibodies. As you can see on the right, inclusion of anti-human Vista antibodies, but not mouse or sheep IgG control antibodies, completely blocked this interaction between Vista and VSIG3, suggesting that VSIG3 is a novel binding partner for Vista. The biological functions of VSIG3 are still unknown. In an effort to discover the potential functions of VSIG3, we treated human PBMCs with plate-coated recombinant VSIG3 protein in the presence of anti-CD3 antibody to mimic T-cell receptor ligation. Interestingly, immobilized VSIG3 protein significantly inhibited anti-CD3-induced IL-17, RANTES, MIP1-alpha, and ITAC secretion from human PBMCs. The cytokine and chemokine profile in cell culture supernatants was determined using the proteome profiler Human XL cytokine array kit. To confirm the cytokine array data, PBMCs were treated with varying concentrations of plate-coated VSIG3 protein in the presence of anti-CD3 antibody. VSIG3 treatments inhibited IL-17 secretion in the cell culture supernatant. IL-17 levels were measured with the Quantikine human IL-17 ELISA kit. To further confirm the cytokine array data, PBMCs were treated with varying concentration of plate-coated VSIG3 protein in the presence of anti-CD3 antibody. Consistent with our cytokine array data, VSIG3 significantly reduced production of anti-CD3-induced RANTES, MIP1-alpha, and ITAC from human PBMCs in a dose-dependent manner. RANTES, MIP1-alpha, and ITAC secretion in the cell culture supernatant was measured using Quantikine ELISA kits. Next, we evaluated whether or not VSIG3 VISTA interaction mediates the VSIG3 immunosuppressive effect. To this end, we treated human PBMCs with plate-coated VSIG3 protein in the presence of anti-CD3 antibody and various concentrations of human recombinant VISTA protein. On the left, you can see that soluble VISTA protein pretreatment abrogated VSIG3-induced RANTES inhibition in a dose-dependent manner, indicating that the effects of VSIG3 on RANTES inhibition is through the VSIG3 VISTA pathway. Furthermore, we incubated anti-human VISTA antibody pretreated human PBMCs with plate-coated VSIG3 protein in the presence of anti-CD3 antibody. The anti-human VISTA neutralization antibody pretreatment significantly attenuated VSIG3-induced IL-17 inhibition in a dose-dependent manner. Taken together, these data support our hypothesis that VSIG3 is a ligand for VISTA, and the VSIG3 VISTA interaction has profound co-inhibitory functions on human T-cell responses. Next, we wanted to test the effects of VSIG3 on human T-cell activation. We isolated human CD3 positive T cells from PBMCs and treated them with plate-coated VSIG3 protein in the presence of anti-CD3 antibody. As expected, VSIG3 inhibited anti-CD3-induced IL-2 and interferon gamma production on human T cells in a dose-dependent manner. Human Quantikine ELISA kits were used to quantify the levels of IL-2 and interferon gamma in the supernatants of human T cells for these experiments. Similar to the previous slide, we isolated human CD3 positive T cells from PBMCs and treated them with plate-coated VSIG3 protein in the presence of anti-CD3 antibody. As shown on the left, VSIG3 also inhibited anti-CD3 induced IL-17 production on human T cells in a dose-dependent manner. It is clear that VSIG3 inhibits T cell, IL-2, interferon gamma, and IL-17 production, and next we wanted to test the effects of VSIG3 on T cell proliferation. VSIG3 inhibited anti-CD3 induced human CD3 positive T cell proliferation in a dose-dependent manner at 72 hours post-treatment. Human T cell proliferation was assessed with the rosazurin fluorometric assay. The results from these assays on T cells suggest that VSIG3 acts as an inhibitory ligand and directly inhibits human T cell activation. In summary, here we report that VSIG3 is a novel ligand for VISTA and that the engagement of VSIG3 with VISTA on activated T cells inhibits T cell proliferation as well as cytokine and chemokine production. The co-inhibitory functions of VSIG3 on activated T cells, combined with the highly elevated expression of VSIG3 on colorectal cancers, hepatocellular carcinomas, and intestinal-type gastric cancers, suggest that blockage of the VISTA VSIG3 pathway represents a new cancer immunotherapy strategy. We currently offer 7 VSIG family member proteins with human VSIG1, human VSIG8, mouse VSIG1, and mouse VSIG3 all coming soon. Much like the Butyrophilins, many of the VSIG proteins inhibit T cell activation and are therefore promising new targets for immunotherapy research. Next, I would like to turn our attention to an exciting class of immune receptors called the leukocyte immunoglobulin-like receptors, LILRs. Other names for these receptors include immunoglobulin-like transcripts, ILTs, leukocyte Ig-like receptors, LILRs, and CD85s. There are 6 LILR-A family members and 5 LILR-B family members in the human genome. The LILR-A family of receptors generally activate immune responses in immune cells, while the LILR-B family members contain intracellular ITIM domains and generally inhibit immune activation. Blocking anti-LILR-B antibodies or secreted ECD-LILR-B proteins might have strong potential as next-generation immune checkpoint reagents. As you can see from the publications on these LILRs shown on this slide, the LILR-B family of receptors blocks T-cell function and are already being described as future targets for immunotherapy research. It is well known that LILR receptors bind to various receptor ligands including MHC molecules, S100A8, S100A9, CD1D, oligomeric beta amyloid, NOGO66, MEG, and integrins. We became interested in these LILR receptors after coming across a Nature paper by Zhang et al. Published in 2012. The authors of this paper showed that angiopoietin-like proteins can bind to LILR-B2 receptor, and this interaction supports expansion of hematopoietic stem cells. At the time, we had many recombinant angiopoietin-like LILR-A and LILR-B proteins in our freezers, and we decided to do a small-scale ELISA screen investigating if angiopoietin-like proteins bind to additional LILR receptors. We found that indeed, many of the angiopoietin-like proteins do bind to other LILR receptors not described in the Zhang et al. Paper. We next set out to investigate how angiopoietin-like signaling via LILR receptors might be involved in immune regulation. On this slide, we have representative ELISA data showing angiopoietin-like 7 binding to LILR-B4 on the left. On the right, we have ELISA binding data demonstrating LILR-A3 binding to angiopoietin-like 3. Many of the LILR-A and LILR-B receptors are highly expressed on monocytes, so we decided to test the effects of angiopoietin-like proteins on human monocytes. We have found, as others have reported in the literature, that angiopoietin-like proteins are most active as oligomeric complexes, and that these oligomeric complexes are necessary for proper angiopoietin-like function. To aid in forming oligomeric angiopoietin-like protein complexes, we cross-linked the His or MIC-TAG angiopoietin-like proteins with anti-His or anti-MIC cross-linking antibodies. In this experiment, we can see that GM-CSF and TNF-alpha are only induced in human monocytes upon cross-linking of the angiopoietin-like proteins. We do not see any GM-CSF or TNF-alpha induction when angiopoietin-like proteins are not cross-linked. Interestingly, the structurally related angiopoietin-1 protein also was shown to bind to LILR receptors, and it behaves in a similar fashion to induce GM-CSF and TNF-alpha when cross-linked. To control for antibody-dependent Fc receptor activation as a possible mechanism for induction of GM-CSF and TNF-alpha, we ran the cross-linking antibodies alone and detected no induction of these inflammatory cytokines. GM-CSF and TNF-alpha levels were assayed using our proteome profiler human cytokine array kit. In an effort to test if LILR receptors are involved in the induction of immune cytokines in human monocytes, we again tested angiopoietin-like 3 for its ability to induce TNF-alpha. As you can see, angiopoietin-like 3 treatments resulted in a robust induction of TNF-alpha in human monocytes. When a dose range of our secreted ECD LILR-B2 protein is added to a constant dose of angiopoietin-like 3, we see a dose-responsive inhibition of induction of TNF-alpha by angiopoietin-like 3. This data demonstrates that a soluble ECD LILR-B2 protein can bind to angiopoietin-like 3 and inhibit angiopoietin-like 3's ability to induce inflammatory cytokines in human monocytes, suggesting that LILR receptors are the receptors for angiopoietin-like proteins in human monocytes. Here are our product offerings for angiopoietin, angiopoietin-like, and LILR proteins. As you can see, we have almost a complete list of these protein families, and we are currently working to get the remaining proteins on the market in the near future. We have not currently investigated the role of ANGPTL-like proteins in the context of LILR receptors in T cells. We hope that these product offerings will help our customers expand our current knowledge of how these growth factors and receptors are functioning in T cell biology. Now I would like to briefly summarize our findings. We have shown you data today that demonstrates Butyrophilin 3A1 inhibits T cell activation, but the T cell receptor is still unknown. VSIG3 interacts with Vista to inhibit T cell activation. ANGPTL-like proteins bind to LILR receptors and activate innate immune responses in monocytes. LILR B receptors with ITIM motifs are potential new checkpoint inhibitors. Thank you for tuning in for our webinar today. Now I would like to answer any of your questions regarding the data that was presented during this webinar. Have a great day and good luck with your research.
Okay, thank you very much for that informative webinar, Tony, and thank you for sharing your research with us today. I just want to remind the attendees that if you would like to ask a question about any of the research or immune checkpoint inhibitors that were discussed today, you can do so in the Ask a Question box, which is just below the presentation screen. We will begin taking some questions. I see some coming in at this time, and a couple to start, and then we will answer as many as we can, and if yours does not get answered, we will answer it offline and send those back to you in an email. So the first question is on the Butyrophilin family. So Tony, do you know the receptor for BTN 3A1? And if it's unknown, are you guys doing anything to study that? You must be having some technical difficulties here. I don't hear Tony on the other line anymore.
Wait, do you hear me now? Scott?
Yes.
Okay, yes, I heard your question. I'm sorry. We do not yet know the receptor for Butyrophilin 3A1. We are currently trying to figure out what may be binding to Butyrophilin 3A1 in the context of T cell biology, but we do not have an answer for that at this time.
Okay, very good. There's another question, again on BTN 3A1. Is BTN 3A1 expressed on specific types of cancer tissues or cell lines?
Yes, it is. It's expressed on ovarian cancer cells as well as some other cell lines, but not a lot is known about that at this time either.
Okay. Very good. Looks like a couple questions have come in here on the VSIG and Vista section of the talk here. So,
sure.
Is VSIG3 expressed on normal lymph tissues or on naive immune cells?
Not much is known about the expression of VSIG3 at this time. We don't know if it's expressed in normal lymph tissue yet or if it's in any immune cells at this time.
Okay. Another one, does Vista bind to any other VSIGs?
In the ELISA binding screen conducted at R&D Systems, we were unable to see Vista binding to any other VSIGs. Although there are, we did find some patents that did report Vista binding to VSIG8. Although we have not been able to, we did see that. We were not able to confirm that finding here at R&D Systems under the same experimental conditions.
Okay, very good. And then last, another question here on Vista VSIG. What kind of research will you or Bio-Techne be doing in the future for VSIG and Vista?
We're currently further studying this interaction between VSIG and Vista in physiological conditions. We're trying right now to determine if VSIG3 is expressed on immune cells, including dendritic cells, macrophages, T cells, and B cells. We also study downstream signaling of the VSIG3 Vista pathway to determine the intracellular signaling mechanism of how VSIG3 Vista and this interaction is modulating T cell activation. But those studies are currently ongoing.
Great. Looks like a question came in here for LILRs. Did you investigate the role of LILR in NK cells?
We have not at this time. The interest in the LILR receptors, as I discussed in the presentation, is primarily focused on the LILR-B family members. They have intracellular ITIM domains and have been shown to inhibit immune activation in various immune cells. Most interestingly, when the LILR-B family of receptors are activated in T cells, it inhibits T cell activation. So clearly these are becoming a major target for immune therapy research. And the LILR-As, which are likely the receptors involved in the immune activation in the monocyte data I presented during the webinar today, are a little less interesting as far as immune therapy research. The LILR-Bs, particularly in the context of T cells, and the publications we see, suggest that those are the true targets for antibody therapies and such.
All right, very good. So there are a number of questions coming in, actually too many to kind of get to make sure that this webinar stays within a reasonable time length. So I think what we'll do here is we'll end the webinar, and for those of you who asked questions that were not addressed here by Tony right now, I'm going to give him a list of the questions, and we're going to email out the whole list of questions and answers to anybody who attended or registered for the webinar. So you'll get your questions answered then. But I thank you again. I thank Tony for putting on this webinar for us, and I hope you all enjoyed it and it was informative, and look forward to emails coming with questions regarding the webinar and a download link so you can watch this again at your own time. So thanks again, and have a good day.